A very good afternoon to everyone. Myself, Upasana Hazarika, Computational Biophysics Laboratory, MBBT Department, Tezpur University, Assam, India. Today, I am going to present my research work on investigation of Leishmania Donovanis key pathways by comparative network analysis to uncover new
Please
therapeutic
share your
targets.
full screen.
So this is the contents, and the main objective of this study is to common target establishment for multiple pathways by
Please
system biology approach.
share your full screen, please.
We
Yes,
don't
sir,
see
I am
your full
sharing.
screen. Share your full screen, please.
I am sharing, ma'am.
Share your full screen. We didn't see your full screen.
Okay, just 1 minute. Ma'am, is it fine now?
Now we don't see anything.
Is it fine, ma'am?
More or less.
Yes, it's
Okay,
wonderful.
so may I
Continue,
start?
please.
Continue.
Okay, so a very good afternoon to everyone. Myself, Upasana Hazarika, Computational Biophysics Laboratory, MBBT Department, Tezpur University, Assam, India. Today, I am going to present my research work on investigation of Leishmania Donovanis key pathways by comparative network analysis to uncover new therapeutic targets. So these are the contents, and the main objective of this study is to find out a common target establishment for multiple pathways by system biology approaches. Leishmaniasis is a serious health issue impacting 88 nations. Is a vector-borne sickness spread by the bite of female sandflies. There are many clinical forms, among them Leishmania Donovanis caused by the VL, which has been described as the most severe type, which can be deadly. VL has impacted Nepal, Bangladesh, and some other states of India, such as Bihar, West Bengal, and Uttar Pradesh. Increase in cost, toxicity, and side effects of available treatment with the emergence of drug resistance parasite point to an urgent need for new efficient drugs. For this study, I chose 5 metabolic pathways depending upon the which is dependent upon the survival and survival essentiality for the Leishmania Donovanis. These are glycolysis, pentose phosphate, glutathione metabolic pathway, purine metabolic, and pyrimidine metabolic pathway. So finding in common targets could be an effective strategy to eliminate the pathogen and to treat VL. So for this research work, I followed the 4-step methodology. First one is the extraction of the pathways data. For this, I mainly use string and cake databases. Next one is the construction of the network and the subnetwork. For this, I use cytoscape tool. The third one is the topological analysis and the network comparison. For this, I used cytoscape plugin like CytoHubba, MCode, Centiscape, Clubu, and Clupedia. And the fourth step is the structural study of the protein. For this, BLAST and modular is used. Now coming to the results section, first figure represent the Venn diagram representation of common enzymes. This I made 2 groups of the selected pathways. One is 2 pathways from the carbohydrate metabolism along with the glutathione metabolic pathway, and the second group I present the nucleotide metabolic pathways and the glutathione metabolic pathways. So in the figure 2, I showed the constructed network for glycolysis pathway. For this, I found 102 nodes and 496 edges. Similarly, for the PPP, nodes found 70 and edges 301. For the third one, glutathione metabolic pathway, node found 153 and 1002 edges. For the purine and pyrimidine, nodes found 231 and 211 respectively, and the respective edges are 20231 and 1497. Next, I go for a subnetwork construction approach to see the most impactful node of the top 10 node in each of the network. And here the deep color represent the most impactful node, to decreasing color represent the weakness of the intensity in that network of that particular hub protein. So after that, I go here, I represent the number of common nodes that are depending upon the basic centrality measurements like betweenness centrality, closeness centrality, degree, and bottleneck. From the 11 topological measurement, I selected that 4 for my study, and then I found for the glycolysis 6 proteins are common among the top 4 protein top 4 analysis. Similarly, for the other 7, 2, 3, and 2 are found as common respectively. Next, I go for the molecular cluster detection analysis, that is MCODE, and here for the glycolysis, I found top 9 module, and where PGI and phosphoglucomute is found as the most highest degree cluster closeness and betweenness centrality done using Centriscape. Similarly, for pentose phosphate, G6PD, PGI, and phosphoglucomutate are found among the in the glutathione, RNM1 and G6PD found in the purine. Similarly, RNM1 and phosphoglucomute is found in the pyrimidine pathway. RNM1 is found among the 11 module. Next, I go for the gluco analysis to see how many biological process or other processes that 4 particular nodes are actually involved. Gluco, where the GLO reveals the number of genes associated with both terms corresponding to the molecular function, cellular component, and the biological processes. For glycolysis, I found that PGI is involved in PGI and phosphoglucomute is involved 1/3 of the total biological processes. For PPP, we can say that PGI is involved among the PGI is involved 34 processes among 54. Similarly, other 2 is involved 1/3 of the total processes. For the pyrimidine and for the glutathione metabolic pathway, similarly 1/3 of the processes that top genes G6PD and RNM1 involved. Here in the purine metabolic pathway, similarly 1/3 of the processes RRM1 and phosphoglucomutase is involved, and pyrimidine also 18 out of 40 processes RRM1 is involved. So these are the model target proteins. Here PGI and G6PD are the common in between 2 discrete pathways, and phosphoglucomutase and RRM1 is the common protein among three common pathways. So these are modeled using the modeler and validate using the safe server. Now, in conclusion, we can say that this study focused to explore disease-associated pathways, followed by a comparison of networks, and identified common hub proteins by aggregating the behavior of proteins connected in an PPI network. We are proposing 2 common targets from 3 different pathways and another 2 common targets from 2 discrete pathways. The common hub proteins identified here can be further studied as in therapeutic targets. So these are a few selected references I use in my study. Now, I acknowledge my department, my university, and my guide and my lab mates for their advice and suggestion. Thank you so much, BCADD 2023, for providing opportunities to present my research work. Thank you so much. Now, I'm open for discussion.
Upasana, the talk,
Yes, ma'am.
your presentation should be 5 minutes. Keep it in
Okay.
mind. Okay, there is some questions. Thank you for interesting and very important talk, but we have to keep the time. There is some,
Yes.
what is the best similarity? Dmitry Shulga asked you, what is the best similarity to the proteins with known structures to your selected targets used for modeler?
What is the base similarity of proteins in structures?
With known structures for your
Yes,
selected
actually,
targets.
I select the proteins by done MCODE and CLUGO analysis, and because that rather than G6PD, other proteins are not found in any databases. So I model the protein for my further study.
There is other
Hello.
question?
Yes.
Some thoughts, some discussion? Okay, thank you very much for your talk.